Hello, everyone. Can you hear me well?
Yes.
Thank you. Thank you very much. So we have, so what's today, Wednesday? November 4th. So I hope you all voted yesterday. And the votes have been counted. We still don't know. Keeping our fingers crossed for whoever we voted for. All right. So we had our test. You did very well. Much better than on the first test. Some of you sent me some emails. I responded to those. If you still have any questions, let me know. The most common question you guys had was related to the nucleotide storing genetic information. So the nucleotide by itself does not store genetic information. It's the sequence of nucleotides, which is called a polynucleotide, which stores genetic information. Could be considered as tricky, but I don't know, by going by definition, it does not store genetic information. So there are a couple other questions. Phosphodiester bond, right? So nucleotide now, you already looked at the answers. Does a nucleotide have a phosphodiester bond? Can you answer me that question right now? Does a nucleotide have a phosphodiester bond? No? Anybody? Anybody want to answer that?
Yes, it does.
Oh, you see, no, it doesn't.
A nucleotide doesn't have a phosphodiester bond?
No, it doesn't.
Well, it doesn't have the phosphodiester attached to it?
No.
The nucleotide does.
What is diester? I mean, go by definition again. Diester. What does di mean?
Oh, two. It just has a phosphate group.
It's a phosphomonoester. That's right.
Ah, tricky.
So you see the phosphate
Yeah.
group. So the phosphate, if you think of phosphoric acid, three OHs, you can have a maximum number of esters that you can make using phosphoric acid is 3. So you can have phosphotriester, phosphodiester, and phosphomonoester, right? And so nucleotide will be a phosphomonoester because you replace one of the hydroxy groups with the ribose. And the other two OHs are free. So it's a phosphomonoester.
Yes, it is. Thank you.
All right. I'm glad we got this one straight. All right. All right. So, but these are some overall, I think, guys, you did very well. And obviously, you studied much harder, you were much better prepared, and for test number 3 you'll be even better prepared. So all right, we only have one lecture dealing with chapter 9, and in fact I don't even know we need the whole lecture. The primary reason is this the topic. The primary reason is the topic, which is DNA based technologies. DNA-based technologies. And this, in my view, at least, this doesn't belong in this class. I don't know why this put this chapter in this textbook, because this is really not biochemistry. This is molecular biology. This is molecular biology, and you will study this in other classes, primarily taught by biology professors. There are some chemistry professors who also work in this area. But certainly an organic chemist such as me should not be teaching you this, since this is not what I do for a living. And my research has nothing to do with this. And it's not molecular, right? So what biochemistry is about is structure of molecules and how that relates to biological processes in a body. So DNA-based information technologies does not deal with structure of molecules. Okay. So the most you will see in DNA-based technologies is G goes to C, A goes to T, right? So base pairing, complementary base pairing. Other than that, there's no molecules. There's no molecules, and that's why we're not going to spend much time on this chapter. And but since it's covered in this textbook, we are going to respect that and spend one lecture on that, or maybe even less. I might be even let you go before the end of this lecture. Here's, I'm going to share my screen. All right, so what we are going to do is we're going to talk a little bit about DNA cloning techniques and expression of recombinant proteins. So these are the most common ones for molecular biology. And so, and we will not cover microarray, and we will not cover this. So just these two. And recombinant DNA. So recombinant DNA, one thing is to remember from this whole word of recombinant is that it's artificial DNA. Artificial DNA does not exist in nature. And basically it forms the basis of the modern molecular biology, right? So when somebody says recombinant DNA, that probably means molecular biology, and specifically means cloning. Cloning is something that we're going to talk about in the next 15 minutes. We'll talk about overexpression of proteins. So many proteins are not easily accessible through their isolation from the particular cells or organisms. And so they have to be synthesized. And so we talked about biosynthesis of proteins, but probably the best way to make proteins is to make biological systems synthesize them for us. And so this is known as overexpression of proteins, right? So we're going to use, for example, E. coli. We're going to use bacterial cells to make proteins for us. And transgenic foods and animals. So you already know some about this. In fact, that has to do with organism cloning. Organism cloning, you have heard of this through multiple sources, right? So basic creation of genetically identical copies of an organism. Right, so basically you take a nucleus from one organism, you put it in the egg cell of another organism, take the nucleus out of the egg, take a nucleus from the somatic cell, put it in, and put it in the mother who will deliver progeny, right? So you know that many organisms have been cloned at this point, right? So the original one was a sheep in Scotland, but nowadays we have a lot of organisms that have been cloned. A lot of this has ethical issues, right? So you have heard about human cloning, and human cloning has a lot of ethical issues to deal with. So now we will talk about DNA cloning. So organism cloning is the cloning of the whole organism. So it's a very complicated process. Now for DNA cloning, it's easier, right? So basically we're just gonna make identical copies of a piece of DNA. And we're gonna basically take a specific gene from a source organism and amplify it in a target organism. So the way it's done, so the basic steps are, we're gonna cut the source DNA from the organism that we're interested in. We'll select a suitable carrier, often called as a vector, right? Vector, we're gonna insert this vector, or we're gonna use the vector to insert the gene into the target organism, where we're going to amplify our DNA. So oftentimes referred to as host cell, right? So we're going to insert this recombinant vector into host cell and let the host cell produce multiple copies of recombinant DNA. So sounds very simple, and in fact, it's actually it is pretty simple nowadays. Something that molecular biologists do on a daily basis. So so how does it work? So in step one, we're going to generate recombinant vector. So you know the DNA in prokaryotic cells exists is in chromosomal form, as well as in the form of a plasmid. So plasmid DNA, as you learned in biology classes, is a circular DNA, right? Circular DNA, and it's perfect for, and it has its own it can be reproduced by itself, right? Replicated by itself. And so without the chromosomal DNA, it's replicated separately. And so it represents as a it can serve as a nice vector for the insertion of a foreign DNA into a host cell. So what we're going to do, we're going to take a eukaryotic chromosome with this red gene that we want to be cloned, right? So this is going to be DNA fragment. We're going to cleave it out. We're going to take it out of the chromosome using an enzyme known as restriction and the nuclease. Restriction and the nuclease. And so we're going to use this enzyme to cleave it and take it out of the chromosomal eukaryotic chromosome. And we're going to use the same enzyme to break the plasmid to generate this nick, in the DNA, right? And then we're going to mix these two together, and we're going to use an enzyme known as DNA ligase. DNA ligase, which stitches or which puts the two DNA fragments together, and that will generate recombinant vector. You can see what's happening here because we're going to use exactly the same enzyme. Exactly the same enzyme will cleave this DNA in the same way as it will cleave this DNA, right? So you can see that these sequences here and these sequences here are identical. Identical. And because they're identical, they can be put together by the DNA ligase into recombinant vector. So what do these enzymes do? Basically, they cleave phosphodiester bonds at specific sequences. We just talked about this. Very common in bacteria. There are over 1,000 of them in bacteria. Now, their primary role in bacteria is that they actually allow for the bacteria to fight infectious viral DNA, right? So if bacteria have been exposed to viral DNA, they can recognize specific sequences in the virus, right? And so if they do that, then the infectious virus DNA can be cleaved. And if you cleave the viral DNA, the virus cannot be infectious anymore. All right, so these restriction endonucleases, they can make what's known as sticky ends or blunt ends. That will be on the next slide. We're going to look at this in a moment. And many of these are known and commercially available. Now there's a database. There's a database of those. And depending on what kind of DNA you want to cleave, you go to this database and you find the restriction endonuclease that you want, depending on the sequence that you're looking for. All right, so if this is the chromosomal DNA, so this is the cleavage site. You can see here. So for example, this restriction endonuclease known as EcoR1. So this comes from E. coli. And you can see here there's a sequence here, GAATTC, right? So this sequence, whenever the sequence occurs, the restriction endonuclease will recognize that and will cleave the DNA, forming what's known as sticky ends. Why they call sticky is because now we have fragments of single-stranded DNA, which can form base pairs with another piece of single-stranded DNA, right? And so that's what they call sticky ends. Now on this side, so there's this enzyme, which is, now I don't know how to read this, PIVOL1 restriction endonuclease, which will generate blunt ends. And then you have a DNA ligase, which will bring the foreign DNA and insert it in here, right? So this will be the insertion of the sticky end on this side, and this will be the insertion of the blunt end on this side. And sticky ends are inserted more easily, which makes more sense because sticky ends can preform the double helix, right? And then all you have to do is just stitch up the phosphodiester bonds, basically create the phosphodiester bonds. Now with the blunt ends, it's not so easy, right? So the restriction endonuclease really have to recognize this portion, has to recognize this portion, and the nitrogenous bases do not form base pairs here. So it's much harder to put these together. So sticky ends and the nucleases work much more efficiently than the blunt ends and the nucleases. Any questions? All right. So ligases, enzymes which normally function in DNA repair. So basically we borrow these enzymes from the DNA repair machinery, and it makes sense, right? So when the DNA is cleaved, so there are these ligases which which sew the DNA back together. And there's human DNA ligases, there's bacterial DNA ligases. Both can be used as sources of enzymes for us. So we're going to take our recombinant vector that we put together through the consecutive use of endonuclease and ligase, and we're going to put this in the host DNA. So, for example, E. coli, right? Bacteria. And so this plasmid can be replicated by itself, right? So that in a process known as propagation, right? So you can see the single cell, it can actually be propagated in a single cell, or the cell can divide as well. So from a single cell, we have 6 cells here, but you can see here the plasmid by itself can also undergo replication, right? So double process, basically replication of the plasmid as well as replication of the cell, can multiply the copies of can produce multiple copies of recombinant DNA. Recombinant DNA. All right, so the question is, all right, so there are 2 processes here. One is, how do we know that we put the DNA in? In other words, how do we know that we propagate the desired plasmid? And secondly, how do we select for the cells which have taken up the plasmid, right? So first, the plasmid has to take up the DNA, and secondly, the cell has to take up the plasmid. And so how do we select for those? And we use antibiotic selection process for this. And specifically on antibiotics, for example, such as penicillin or ampicillin, that we know that kill bacteria. So here's a structure of ampicillin. Does anybody know the structural fragment that I'm circling right now? What's the name of this structural fragment?
Is it beta-lactamase?
Beta-lactam, yeah. Right, so beta-lactam antibiotics, that's right. So I'm glad you actually said beta-lactamase. What does beta-lactamase do?
I believe it attacks the cell wall of the bacteria.
Beta-lactamase is an enzyme which normally we don't like. Why? Because it makes the bacteria resistant to antibiotics. So in other words, basically what it does, so beta-lactamase, so if you have beta-lactamase here, we're going to open this ring. So this is sulfur. What else is here? Nitrogen, and so on and so forth. Beta-lactamase. Inactive antibiotic. So if the bacteria have developed resistance to ampicillin or penicillin most likely what it has done, it has developed basically it has it's producing this enzyme known as beta lactamase that actually hydrolyzes beta lactams using water, right? And so normally we don't want this. We don't want this because our antibiotics will be ineffective. But in this case, for the purposes of this class, for the purposes of the cloning, molecular cloning, this is actually a good process, right? And so what's going to happen is actually you can insert a gene. You can insert a gene in the plasmid that will give the host bacterium a resistance against antibiotics. And so this gene, in many cases, will be a beta lactamase gene. Right? Beta lactamase gene. And so and if the bacteria are resistant to this antibiotic, it will grow preferentially in the presence of the antibiotic, right? So let's look at this slide here more carefully. So let's say we have our plasmid. We have a plasmid isolated from E. coli, right? So we have actually two resistant genes. We have a gene for ampicillin, which is beta lactam, and we have a gene for tetracycline. Now it's a different type of enzyme, which makes the bacteria resistant to tetracycline. So we're going to cleave this plasmid with restriction endonuclease and cleave it specifically at this site where ampicillin is, where the ampicillin resistant gene is, okay? So in other words, we're going to disrupt the ampicillin gene, and tetracycline gene will remain intact. We're going to ligate the DNA of interest that we want to be to clone, right? And we're going to transform the E. coli cells with this plasmid, right? So some of these will take up the plasmid, some of these will not. So these cells took up the plasmid, and these cells and these ones did not. You have to keep in mind that for a cell to take up the foreign DNA is not an easy process. Why do you think cells don't take up DNA so easily? Why the DNA cannot just simply go inside the cell and find home there? Just think about the cell membrane and the structure of DNA that just as we talked about in this class. Phosphodiester bonds.
Isn't DNA, doesn't it have a strong like negative charge and it would have to pass through that phospholipid bilayer? Nice,
yes. So this is your phosphodiester. The two riboses on either side, this is diester. Yeah, something that you got tricked on this test about. So there's a phosphodiester and there's a negative charge. There's a negative charge, it cannot cross the cell membranes very well. So the DNA can, and the plasmid as same thing, right? It's just a circular DNA, cannot cross the cell membrane very easily. And to transform a cell, there are some special procedures have been developed. One of them is known as a heat shock. Heat shock. Another one is known as electroporation. Heat shock, basically you just like raising the temperature very quickly. In the electroporation, you apply a electric pulse, which will break up the membrane for a short period of time, right? So the membrane is continuous, but if you apply the membrane just for a second or so or two, the membrane can come apart and the DNA can slide through, right? So the process is known as electroporation, which allows for the DNA to slide through the membrane, which had the electric pulse applied to it. All right, so here's our DNA. Plasmid has gotten into some cells, and then what we will do is we're going to select for those cells. We're going to select for those cells by applying tetracycline. So tetracycline, so remember all of these plasmids, so tetracycline is this blue, I think, if I remember correctly, yes. So tetracycline gene for resistance to tetracycline, right? So the cells on the agar, the cells which will grow are those that have the tetracycline resistance gene, right? So if you if you put the tetracycline into the media, then the cells which do not have the plasmid will be killed by tetracycline, right? So the cells which do have plasmid will be resistant to tetracycline and produce a colony. Will produce a colony, and then so once we have selected for those cells, then we actually start growing colonies of cells which have acquired the gene of interest. So specifically, now we're going to put the in the media. Not only we're going to put the tetracycline, we're going to put ampicillin. So this is going to be our control. And so let's look at this. Let's look at these two agar plates. And so let's say this colony here does not have the corresponding colony there, which means is that this colony here was killed by ampicillin, right? And this colony here is growing, which means that this is the colony that has our gene, because the ampicillin resistance gene has been disrupted by the gene of interest that was inserted by the ligase. Does that make sense? Here is our restriction and the nuclease, and remember here ampicillin, right? Ampicillin resistance gene was disrupted here, right, by the insertion of the foreign DNA. And so here ampicillin basically when it is disrupted, it is basically kill those cells, right? And here it did not kill those cells because the resistant gene is still there. All right, so basically by comparing these two, you know where the DNA was inserted and you know where DNA was not inserted. And so so it's a double selection process, and that's how you identify these empty plasmids. All right, so these cloning vectors that we just talked about, so these are basically these are plasmids. Circular DNA molecules can replicate autonomously, right? So allows for cloning of DNA up to 15,000 base pairs. Now we'll talk about this bacterial artificial chromosome and yeast artificial chromosome in a moment. So these are utilized to clone DNA, much longer pieces of DNA, up to 300,000 base pairs. So here is going to be the cloning vector. So this is the recognition sequence. by the endonuclease, right? So eco R1 is going to cleave here. Now there are a couple other restriction endonucleases that can cleave at different positions. So this is the tetracycline resistance gene. This is ampicillin resistance gene. Now this is what's known as the ORI, origin of replication. This is actually where the cloning vector will start replicating inside the host bacterial cell. It will start here and go all the way around. Go all the way around. So here's bacterial artificial chromosome. So you can see here, so this is the vector. Now there's also a couple recognition genes, right? So for example, this one is colorimetric one, right? So if this gene is not disrupted, then basically it for LAC-Z, if this gene is not disrupted, it actually is responsible for the production of an enzyme known as a galactosidase. Galactosidase. So in other words, basically what you have is you have this X, which is a dye. And it's linked to galactose. So this is colorless. Now, in the presence of the enzyme, so this enzyme, galactosidase, it will cleave the galactose from the dye and generate the blue color. Plus galactose. And so it's another way to basically select for cells which have the gene inserted in them, right? So now based on what I just told you, so the cells which have the gene inserted in them, will they be blue or colorless? Blue or colorless? Answer my question. Blue or colorless? So the cells which have taken up the gene of interest that we're going to clone, right? Are they blue or colorless?
Blue.
Think about it.
Are they colorless? Because in the image, it looks like it's disrupting it, like it's opening it up right at the.
That's right. Yes.
So yeah.
That's what I think too. Yeah. So so if this produces the enzyme galactosidase, right? If you disrupt this gene for galactosidase, then the galactosidase is absent, right? If galactosidase is absent, then you will not cleave off the galactose from the dye, and the cell will remain colorless, right? And if the cells that have not taken up the gene of interest will have undisrupted galactosidase gene, right? which will, if you administer this dye to them, will cleave off the galactose from the dye, and they will turn blue. Right. All right. So so this will be the bacterial artificial chromosomes. So there's a special vector which is used. So this vector is used, and now there is an additional. So it's actually not a simple process. You actually have to pack this thing together, and you have to choose the genes that will do the job for you. For example, these F plasmid PAR genes. It's not just any genes here. These genes allow for the for when the cells divide, for the distribution of these plasmid in such a way that there are only few plasmids go into each daughter cell. Just think about it. So if this system is designed for cloning of large pieces of DNA, you don't want many copies of plasmid in a cell, correct? You want very few copies. You want very few copies so that there is no DNA exchange. There's enough room, not a room, even important, right? So there's enough room for this plasmid, lone plasmid inside the cell. So you have to limit the number of these plasmid inside the cell, right? So you have to slow down their replication. And so these genes, that's what they're responsible for. Basically, they're responsible for keeping the number of these artificial chromosomes low inside the cell. All right, so there will be restriction in the nuclease. You insert this large DNA fragment. Here it is, recombinant back. We're gonna obviously do this through electroporation. This large piece of DNA will not simply get inside the cell by itself. You have to use special techniques, and electroporation is one of them. So you get this DNA inside the cell, and then obviously then you're gonna do selection of the, first of all, you're gonna do selection of the what's antibiotic starts with a I forgot the name of this antibiotic. Camptothecin? Is it camptothecin? Antibiotic that starts with the C and M. I think, hold on a second. I forgot the name of this antibiotic. Anyway, doesn't matter. So this, the principle is more important than the particular antibiotic. So there is a specific resistance gene to this antibiotic. Oh, chloramphenicol. There we go. Chloramphenicol. Just get back to me. Chloramphenicol. Chloramphenicol. And so so there is basically the gene that will confer a resistance to the cell, to this antibiotic, right? And so you select for those cells, and then in the agar, once you selected those cells, you can see the blue ones are the ones that do not have the gene, right? And the white ones are the ones that actually have both that are resistant to chloramphenicol and resistant to this dye that not resistant, but that makes the dye blue. All right. Any questions? So these are bacterial artificial chromosomes. So you can see here that for selection you can use obviously antibiotic media, or you can also use the color. right? So basically you apply few cells here and they will grow into a colony. And if the colony is blue, that's not what you want. If the colony is white, that's what you want.
I have a quick question on the last one. There was like the double antibiotic resistance, but I guess on this one it's not needed because like the two test, one is the color and one is the CAM antibiotic. Is that it?
Yeah.
Okay, got it.
Yeah. And now you can also do this with yeast. All right. So yeast is a multicellular organism, but sorry, it's a not multicellular. The yeast is a what was eukaryotic? Sorry, eukaryotic. I was looking for eukaryotic. So yeast is a eukaryotic organism, but you can still do this with the yeast as well, just like with bacteria, with E. coli. So here is the technique that has been developed for the yeast artificial chromosomes. Now there's, you can see there's various markers as well. So there are the things to keep an eye on are the selectable marker X and selectable marker Y. There is a telomeres. Telomeres, we talked about telomeres regions in DNA where we have, instead of double helix, is a quadruple. Remember we talked about that, telomeres? When you have Hookstein, remember that word, Hookstein base pairing? Hookstein. When you have the N7 position of purine involved in hydrogen bonding, N7, right? H bonding. So, so these are telomeres. Usually you can make specific sequences telomeric by adding more guanine, G-rich. So if you have a lot of guanines in the, your single strand, then these things spontaneously can form telomeres. Okay, so they can actually form the N7 hydrogen bonds. And so, all right, so here what we're going to do is we're going to cleave the plasmid at these two positions with this restriction and the nuclease, right? Turn this into linear DNA. Then we are going to use another restriction and the nuclease from the E. coli, EcoR1. We're going to cleave at this position, I guess, right? At this position. And then that actually separates the X selectable marker from Y, right? So let's say if your selectable marker, if this produces some kind of color and this produces some kind of color, you know that once you have ligated, the two colors will be produced at the same time, right? So in other words, you know that X and Y have been united together into a single DNA molecule because there are selectable markers for X and Y. All right, so and then that you digest the cell wall enzymatically, allow for the DNA, for the plasmid to go inside, and then you select for X and Y, and this X and Y will tell you that the clone that you're dealing with has the DNA in question in it, right? So the DNA has, if DNA has been put inside, then X and Y have been combined together. All right. So obviously you can use the same technology for site-directed mutagenesis. This is very complex word for something that is very simple. Basically, you want a, let's say you want a piece, you want to clone DNA, that has a specific mutation in it, right? So for example, you want a protein that instead of one amino acid has another amino acid, and you know that the codes come in 3 nucleic bases, right? So you basically put the a different code in your clone DNA, and then this DNA will be cloned and will be ready for the production of a protein with the that has been mutated, right? So all right, that's really all I wanted to say about that. Okay, so so once you have the this is what I want to say about DNA cloning. Now let's talk a little bit about gene expression in bacteria. So so gene expression, basically that all that means is production of protein of your choice, right? So so the technology is similar. You rely on microorganisms to produce a protein of your choice. The major problem is that eukaryotic genes, if you cannot use the messenger RNA, which has been produced from the cells, and primarily because of the introns, right? So eukaryotic genes have exons, coding genes, coding regions, and they have introns, non-coding regions. And so these introns in eukaryotic genes, they pose problems, right? And so specifically because bacteria cannot basically take out these introns from coding DNA, right? And so basically what we need to do is, before we can express a gene in bacteria, we have to prepare a messenger RNA which is intron free, right? So we have to feed the bacteria with intron free messenger RNA before we can produce a particular protein. And so how that is done, remember, many RNAs have single genes. We call these monocystronic. Their messenger RNAs have multiple genes. So we talked about that. They're called polycystronic. And so, how is this messenger RNA produced? It's produced through the construction of what's known as complementary DNA. So cDNA stands for complementary DNA. Messenger RNA is extracted from eukaryotic cells. Now, keep in mind that something that is very useful for us is messenger RNA molecules have polyadenine tails. And that's actually very useful for us in purification of messenger RNA, right? So for example, if you have a bead, and you have an oligonucleotide that is poly-T. So then poly-T can help you isolate poly-A, right? Can help you isolate poly-A, and you can purify poly-A much easier. All right, so so you isolate messenger RNA from eukaryotic cells, and you then synthesize a DNA strand using this messenger RNA using an enzyme known as reverse transcriptase. Reverse transcriptase, basically an enzyme which will allow you to synthesize complementary DNA from RNA. And that will result in a DNA in a hybrid. And the hybrid, what is the hybrid? Basically shown here. So here is the messenger RNA. Let's say this is messenger RNA with the poly-A tail, right? So using this as a template, then you can synthesize the hybrid. You can see here the primer, which is a poly-T, right? Oligo-DT. Oligo-DT will allow you the synthesis in a 3' direction. This will be the messenger RNA DNA hybrid. Messenger RNA DNA hybrid. You also remember that RNA can be hydrolyzed, right? Remember, RNA is susceptible to base. So you can subject the hybrid to high pH. High pH, and the base will destroy the RNA in the presence of DNA, right? So we talked about that. We talked about the fact that DNA is more stable than RNA. So these hybrids can be easily hydrolyzed to give just the DNA. And then you basically use DNA polymerase, use DNA polymerase, use a primer to to synthesize a double-stranded duplex DNA. And so this will be the complementary DNA. So, so whenever you see cDNA, that means a DNA which does not have sequences which code for introns. So only exons, right? So this is so this double-stranded DNA is ready for expression in bacteria. So you can express this clone gene. So there are special plasmids called expression vectors. And so these expression vectors, now there's a difference. They have a lot of other stuff on them compared with cloning vectors. They have, let's see, what do they have? They have promoter sequences. They have operator sequences. They have other genes. Basically, they have genes which will allow for the now these promoter sequences allow for the binding to. So here's the ribosomal binding site, right? So this gene will be responsible for the ribosome binding site. So the construction of these expression vectors is a little more sophisticated, but you can put this together, and you can put all the information required for the expression of your protein, right? And there is also selectable markers, non-genetic markers, for example, antibiotic resistance, just like we talked about. Now, once you synthesize the protein of interest in the bacteria, for example, then how do you purify that? So obviously there'll be thousands of proteins synthesized by bacteria. So there are a number of tricks you can use. There is what's known as a histag, commonly used, but this one is also commonly used, glutathione S transferase. So GST tag. So basically this is a fusion protein. It's a fusion protein. That has protein of interest plus GST, and GST has a affinity for glutathione. So we talked about protein purification before, and so let's look at this in some detail again. So let's say this is the gene for the target protein, right? And this is the gene for GST, for glutathione S transferase. So we're going to combine these two to allow for easy purification for us, right? And this is the fusion protein which we're going to express in E. coli. So the fusion protein will contain the protein of interest, which is the red one, and will contain the GST in it. Okay? And so we're going to express this fusion protein in our bacterial cell. And you can see here, so the red one is the protein of interest, and this one with the mouth open, sort of the mouth open, is the GST, which will bind the ligand, which will bind glutathione. We're going to prepare cell extract containing the fusion protein, right? And there'll be lots of other proteins in the mixture. We're going to add this protein to the column. And the column, you can see here, so this will be the beads. This will be the beads, which will contain the GST tag. Okay, so I guess the red ones, the red spheres are glutathione. Glutathione are the spheres which will bind, which will fit into this mouth here, right? Which will fit into this mouth, and they're basically affinity. This will be affinity purification. So we're gonna put these beads on a column, create the stationary phase, and allow for the protein mixtures to go through. Then the proteins will go through. Most proteins will go through without binding to glutathione, but our fusion protein will bind to the beads because the glutathione GST will bind to the ligand, right? And then once that has been done, the fusion protein will remain at the beginning of the column, right on top of the column. And then we're going to apply free glutathione. We'll apply free glutathione, which will compete with the beads and what will allow for the fusion protein to go through. And the fusion protein will be eluted by the glutathione solution. So this is how the recombinant genes are often purified, by creating these fusion proteins. And I think, do you have any questions about this? All right, so this is, and the last thing I wanted to mention, there's this one single slide I wanted to show you. Which sort of has to do with the previous chapter, but it's still it's a DNA-based technique, so it kind of it was discussed in the textbook as part of chapter 9. So there is a polymerase chain reaction. There is what's also known as a quantitative polymerase chain reaction, qPCR. So whenever you see that, so what that means is that you can monitor how much of the DNA has been produced by the polymerase chain reaction. And so specifically what you will do is you will have a what's known as a fluorophore. Fluorophore basically is a dye. It's a molecule that emits light upon irradiation with light. Okay, upon irradiation with light, it will emit light of a different frequency. And so if you have a hairpin that looks like this, that has a fluorophore on one end and has a quenching molecule on the other end, so the when the DNA base pairing, when it forms, the quenching molecule is found in close proximity with the fluorophore, right? And so when the fluorophore is excited with light, it doesn't emit light anymore. So it's dark. It's dark, you have no light, and that is a signal basically by itself showing that there is no complementary DNA present. If there is a complementary DNA and specific amount of it, which is produced by polymerase chain reaction, it will compete. It will compete for base pairing with the sequence shown here. And you can see here, this is target DNA, right? And we're gonna form base pairs, new base pairs. You're gonna separate the fluorophore from the quenching molecule. So the quenching molecule is no longer near the dye, right? And so the dye lights up. It lights up and produces fluorescence. And you can see here. So what's shown here is that, let's say this is the amount of DNA which is made, right? So for example, here, polymerase chain reaction cycle number. And so you can see here, so let's say we've made let's say 20 cycles of PCR, certain amount of DNA has been made. And this is the fluorescence signal that goes up. And the reason why it goes up is because the fluorophore is being separated from the quenching molecule, right? And then as you continue adding more of the PCR cycle numbers, more of the DNA has been made. And more of the signal will be obtained because more of this quenching molecule will be separated from the fluorophore, right? That's why it's called a qPCR, quantitative PCR, because more of this DNA, the more cycles of PCR you have, the more DNA is being made, and more DNA becomes competitive with the hairpin. And the fluorophore becomes fluorescent. It's actually a very neat, very nice assay, which allows you basically to tell you, which allows you to figure out how much DNA you have synthesized through polymerase chain reaction. That's why it's called quantitative PCR. All right, I think this is pretty much what I wanted to tell you today. And
This is only 9.1 that we need to read in our book for this section.
right, so basically you read about, maybe I'll send you the pages. Yeah, so will be DNA cloning.
Yes, thank you.
DNA cloning and the gene expression, the protein expression. Sounds good?
Thank you.
All right. So I will have a good weekend, right? Keep your fingers crossed for your president, and I'll see you on Monday.
Thank you.
Bye.
Thank you.